Hello, my name is Madison Mond, and this is my local expert project. Today I will be talking about methylation sequencing. What is methylation sequencing? So it is also known as bisulfite sequencing. That's how it's referenced in most of the articles and data that it's used for. What it is it is another approach to next generation sequencing, and it's specifically used to study the methylation patterns of DNA. Methylation patterns are very important markers in the fields of epigenetics and oncology. For example, epigenetics is we have a set of genes, and sometimes they can be turned on and sometimes they can be turned off, and methylation is what does that. So specifically, this approach uses hybridization probes to capture and then enrich the relevant regions of methylation that you want to use in the study. How does it actually work and able to tie into what we know about next generation sequencing and what we've talked about in class? So it starts with taking your sample DNA and running it through a bisulfite conversion, which detects unmethylated cytosines in the DNA. Then it takes those unmethylated cytosines and it changes them to a uracil during your library preparation. And so these are now your converted bases. So after you've gone through your next generation sequencing, these converted bases can then be detected later as a thymine in the sequencing data, and then you can use that to determine the percentages of methylated cytosines. This is just an image to illustrate what I talked about on the previous slide. So if you look here, you can see this allele 1 is methylated. So what it does is it methylates the cytosine here. And then this allele 2 is unmethylated. So where that C was methylated, this one's not. It goes through the bisulfite treatment, and what it does is it takes all of the other C's and turns them into uracils, but except that methylated C stays a C. For this one, all of the C's turned into uracil. So when it's able to convert, this is what we get, and we still have the methylated C here, which is a U here. Then it can go through non-methylated specific PCR and methylation specific PCR. And what that gets you is the differentiation of bisulfite generated polymorphisms, and it also can get you your percentages of these methylated Cs to see how many you actually started off with. This image here is just a general workflow of how methyl sequencing works. So like I said, you have your DNA sample, and then during your library preparation, that is when your DNA will go through the bisulfite conversion. And then you run your NGS, just like we've talked about in class, and then you get a data analysis out of that, and then hopefully you can publish that data that you are getting from your sequencing. This ties into the module. This ties into module 2, where we talked about next generation sequencing. And so we talked about a lot of different approaches to next generation sequencing. And so what this approach does is it just adds yet another step in the library prep. And then you can run your different modes of NGS like we talked about in class. That's going to best suit your data and your time frame and your cost effectiveness. There are a few known limitations on methylation sequencing. So the first one is that the read lengths of 50 to 500 base pairs are the reads that are supported. Sample sizes smaller than 200 databases are recommended, but there is no guarantee that a sample that has greater than 200 databases will give an accurate result. You also must use a reasonable input size just to get the correct amount of coverage that you're looking for. The maximum input of samples per each analysis is 96 samples. And then lastly, if you have a mixture of single N and paired N inputs, this data will not be supported with methylation sequencing. This slide, I just wanted to talk about one other comparable technique to methylation sequencing and then just some advantages of methylation sequencing. So the only other actual comparable technique is called EM sequencing, so enzymatic methyl sequencing. So what this does is it uses enzymes rather than the bisulfite that was used prior. And from looking at the literature, this technique is actually starting to be used more than methylation sequencing because it's giving higher coverage and it's also giving much more accurate methylation levels than methyl sequencing. But some of the advantages of methylation sequencing is that you can discover patterns of CpG, CHH, and CHG regions across the entire genome. And then one thing that I found really cool was that you can view methylation at almost every cytosine in a genome, with most species using whole genome bisulfite sequencing. There are a few controls used for this technique, and I found most of my information from Thermo Fisher, and all these controls can actually be bought from Thermo Fisher. So the first control is a reference guide DNA that is nearly completely methylated at the CpG sites. And then on the other end of the spectrum, there's also reference guide DNA that is completely unmethylated at all CpG sites. And then right in the middle, you have a native control guide DNA with as-is methylation, which is basically it has a mixture of methylated and unmethylated guide DNA just to give a more normalcy kind of right in the middle control. Moving on to the figures from primary research articles, this was one of the first ones I found, and it's out of an article that's titled The DNA Methylation Landscape of Chinese Hamster Ovaries. And basically what we're looking at here is the global distribution of methylation levels. And so the main one I wanted to focus on is this A figure right here, which is looking at hypomethylation, so undermethylation, partial methylation, or hypermethylation. And so this is exactly what this technique will pop out, and then they were able to use this within their research. So this is the figure 2 that I found from a different primary article, and it's titled Genome-Wide DNA Methylome Variation in 2 Genetically Distinct Chicken Lines Using Methyl-seq Sequencing. And so this whole image is about the global profile of chicken DNA methylome, and the main one I really want to focus on is this A figure right here. And so what we can see here is the percentage of methyl cytosines identified in chicken lungs, which is really what I talked about at the beginning, is that is what Methyl-seq is testing for, and that's the data you're going to get spit back out, is how many methylated cytosines did you have present in your DNA. My figure 3 that I found from the third primary article, and this one was from a paper called CpG Methylation Patterns of Human Mitochondrial DNA. And so what these graphs are looking at is the comparison of mean methylation levels between linear mitochondrial DNA group and circular mitochondrial DNA group. So what they did was they found the methylation levels at these certain sites, and then they were able to compare them between the circular mitochondrial DNA and the linear mitochondrial DNA. And that is the end of my presentation. All my references are on my slides, and thank you very much for listening.